How does the immunological synapse work in immune cells? Imagine two immune cells meeting at just the right spot to exchange vital signals. This special contact point is called the immunological synapse. It's like a tiny communication hub that helps immune cells work together. When a T cell, a type of immune cell, encounters an infected cell or a cell showing a threat, it forms this organized contact. The T cell has receptors called T cell receptors that recognize specific antigens, which are pieces of pathogens or abnormal cells. These antigens are presented on molecules called major histocompatibility complex molecules on other cells. Once the T cell receptor binds to the antigen major histocompatibility complex, the immunological synapse begins to form. This contact is very precise and involves the clustering of receptors and signaling molecules into specific patterns. The synapse is very narrow, about 15 nanometers wide, which keeps the cells close and allows for effective communication. The synapse is made up of three main parts: antigen receptors, adhesion molecules, and co-stimulatory or checkpoint receptors. Adhesion molecules act like glue, holding the cells tightly together. Co-stimulatory and checkpoint receptors help decide whether the T cell should activate or stay inactive, preventing overreaction or autoimmune responses. Inside the T cell, the formation of the synapse causes the cell's internal skeleton, made of actin and microtubules, to reorganize. This polarization directs the cell's centrosome, which is like its control center, toward the synapse. This movement helps deliver signaling molecules and vesicles directly to the contact point. These vesicles carry additional T cell receptors and signaling proteins that amplify the activation signals. The synapse also acts as a control station for secretion. The T cell can release cytokines, which are signaling proteins, or cytotoxic granules, which contain enzymes to kill infected cells. This focused delivery makes the immune response more effective. Natural killer cells, another type of immune cell, also from immunological synapses. They use these contacts to deliver enzymes that kill infected or abnormal cells. Their synapse formation is regulated by activating and inhibitory receptors, making sure they only attack harmful cells. The immunological synapse is essential because it ensures that immune responses are specific and controlled. It helps T cells recognize the right targets, receive the proper signals, and carry out their functions. If the synapse doesn't form correctly, pathogens or tumors might escape detection, or autoimmune diseases could develop. Understanding how the immunological synapse works has helped scientists develop new treatments. For example, some cancer therapies target checkpoint receptors to boost T cell activity. The synapse acts as a signaling hub and secretory site, making it a key focus for immune-based therapies. In simple terms, the immunological synapse is a highly organized, dynamic interface that helps immune cells recognize threats, communicate, and respond effectively.